This video is going to focus primarily on the scientific method and practicing science, but in light of biology students who are studying life on Earth. And so, just a precursor for the entire history of humanity, people have just been amazed by the wonder of life around us. There is so much of it, but obviously we would like to understand, right? And so we see life everywhere. There is, the large obvious life, but then also inside of us, we realize there's this microscopic world, not only our cells, but there's all of these bacteria which live inside of us in our microbiome. These bacteria are so numerous that literally they are the most abundant organisms on Earth, and there's even more viruses. So even taking a tiny milliliter of water from a typical sample, just a milliliter of water has all of this life in it. Viruses are not technically alive, although one could make an argument for some of the larger ones. But viruses do have many of the features of living things. They seem to form related groups. They seem to evolve over time. And so the evolution of the group that includes coronavirus, not only MERS, but then SARS coronavirus 1 and 2, certainly of interest. When we look at bacteria, we see some of them are performing photosynthesis, some of them are decomposers, some of them are parasites, some of them are tiny, some of them are large enough to see without a microscope. They're actually visible to the naked eye. Some of them are harmful to us and causing diseases, but then others are putting oxygen into the air and recycling the nitrogen, which we so desperately require. So, there's this, great diversity here. There are other organisms, which we often refer to as protists, which can be one-celled. Some of them are so small that they're actually smaller than some bacterial cells, but others can be multicellular and then even form brown algae, which can be more than 100 feet long in the kelps. There's this great diversity here. Some do photosynthesis, some include parasites, some live inside the bodies of others, some are predators, some are decomposers. And so there's this amazing, diversity here. There are then three groups which are either multicellular or primarily so, one being the fungi. Fungi include yeasts, which are unicellular. But then there's more than 100,000 species of fungi. Some include the largest organisms on Earth because their microscopic mats of these tiny strands can spread over 2,000 acres. A 2,000 acre single individual. That's a huge living thing. Some fungi cause diseases. Most are decomposers. Some cause diseases in animals, some cause diseases in plants like molds and mildews and rust and smuts. But then some are living inside the bodies of others. So living inside the roots of plants, helping them gain nutrients. Yeast, some of them are obviously very important to humans in the yeast. There are a great diversity of plants alive today. Some are very simple and lack roots, stems, leaves, seeds, and flowers. But then there are some which have vascular tissue, allowing them to grow tall, but do not have seeds or flowers. There are pine trees which have seeds but not flowers. Many flowers are pollinated by animals and thus are very colorful. Many are pollinated by the wind and thus are not as obvious, just because they're not trying to attract animals with smell or obvious visual colors. And then there's this great diversity of animals, more than 1.5 million species that we know of, most of which are the invertebrates. So vertebrates, which are often what we think of first with the animals, these are not the most common vertebrates. It's most common animals. It's actually the invertebrates. Some lack tissues. They're so simple. Some have tissues but no organs. Some are microscopic, as in the microscopic nematode worms. And so there's this amazing, so here's a microscopic nematode worm. There is this amazing diversity of life on Earth. So how do we learn about this? Well, for many people, I think science is something that we do in school, and we learn that all throughout our lives, that, it's presented to us that, so for example, there are characteristics of living things that all living things share. So, put this in a list, and then maybe this is going to be on an exam, so that, they have a complex organization. Even a simple cell is really complex compared to anything in the non-living world. All living things have a metabolism. All living things respond to the environment. They move. All living things reproduce. They evolve, etc. And so for many people, perhaps that's how one approaches, the understanding of the natural world. You go to, school, your teacher presents, something, you memorize a list of things, you spit it back on a test, and that's it. However, science is so much more than that. And so for the next generation of scientific thinkers, for the next generation of biologists, this has to be part of the discussion. So for example, just when we study the world scientifically, it's with the understanding that we don't know the answers yet, all right? And we're trying to find it. So we ask questions and then ask, what does the evidence suggest? Not what do I think, not what do I want to be true, but what does the evidence support? And in the end, that's, how we base our conclusions, not on, the textbook said this, or this really smart person said this, or this, makes the most logical sense to me. It's like, no, this is what the evidence supports. And this other idea, it was great, but the evidence doesn't support it. It seems to be false. We're going to have to modify our conclusions. And certainly as we look through history, I mean, there was most of human history where people felt that spontaneous generation was how many living things were propagated. So if you asked, where did the little fish come from, or the worms, or the bugs, or the little critters, or why does the water turn green when left in the sun, many would feel that that was just life spontaneously generating. Well, that was shown to be false, but for the longest time that was held. For a long time, when people, found big fossil bones, say of mastodons, they would feel that that was, giant humans. There were the beliefs in dragons and giants and unicorns. And so mastodons were interpreted as, giant humans, and the hole for their nostril here was thought to be the hole, for a big eye. So maybe they were a cyclops giant. But that was not true. It was thought that animals could not become extinct. So there was the idea of this great chain of being. It's impossible for living things to become extinct. Thomas Jefferson was a proponent of this idea. That was shown to be false. So in the past, certainly people who have studied the natural world have been wrong. And all of science acknowledges that. All right, so anything that you learn in a science class could be wrong. All right, scientists don't, 100% know anything. Everything is open to doubt and question. And we need to continue asking questions, not just because some of our existing conclusions may be wrong, and they are. A lot of what I teach my students, I'm sure, is wrong. And 100 years from now, scientists will look back and say, it once used to be thought this, we now know better. So not only do we have to correct any errors which we currently have, and I'm sure that we do, but then also there's just all of this stuff we don't know. So for example, if you were to just look at the temperatures increasing with climate change, if this yellow bar is the standard temperature for the 20th century, all of the recent years have been well above average. Temperature is increasing. Well, I just went through all of this diversity of life on Earth. How will increasing climate or increasing temperatures change those organisms? And the answer is, we don't know. Climate change is going to come along with changes in rainfall, patterns, changes in the incidence of severe storms, rising sea level. How will this affect these organisms? We don't know. The human population is growing, and as we need more space for farms and cattle ranches and cities and roads, etc., habitat is decreasing. So how will that affect species? What is causing these couple species given here to become extinct and endangered? We don't know. How will this affect distribution of living things? Will animals have to then be found higher on mountains or farther to the north as the temperature climbs? We don't know. And so this is then a plea to you, the biology student. There is so much that we don't know, and therefore the torch has to be passed. You're the next generation of scientific thinkers. You're the next scientists who help us understand the world better. We don't understand the world we currently live in well enough, and it is changing, and we don't know how. All right. So, for example, here we look out west, and wildfires are becoming more more prevalent. And it turns out it's complicated. Not only is there less rain, warmer temperatures, but also the warmer temperatures are affecting insects like pine mountain beetles, which are now preying on the conifers, which now cause them to die. That adds dead wood. All right, what's happening to the coral reefs as climate change? Well, it certainly seems to be bad, but how will this affect sea life? And so we need individuals, we need you, quite frankly, the next generation, to study this world scientifically. Okay, so let's just begin with a couple of premises with the idea that science is a skill that one develops like any skill, but just through practice. And so, hopefully if you're taking a science class, that this is not just an introductory lecture, like this one, but that also something that can be integrated throughout your semester. So what people do at some level, and what scientific thinkers do, is they ask questions. So for example, if you look at these jellyfish, just to pick an example, you see them pulsating. And a scientific thinker might say, I wonder if they're pulsating all at the same rhythm. I mean, will jellyfish always have the same, 12 pulsations per minute? Or does it vary? Is like the jellyfish responding to stuff that would change that? And so what a scientific thinker might do is, instead of logically think about it, because that's what the Greeks did. All right, so Aristotle, who was perhaps the first true biologist, and all respect to Aristotle. Aristotle said, I wonder what part of the body is the part we feel emotions with. Is it the brain or the heart? And then he reasoned, wait, if I see something that scares me, or if I see someone I'm attracted to, look, my heart beats faster. So logically, the heart is the seat of emotion. That makes perfect logical sense. It's wrong though, but it does make perfect logical sense. So we scientists, we don't depend on, this makes sense to me. This seems logical. I like that answer. That can all be part of making your hypothesis, but science is based on observing the natural world. And so at some point, you then say, all right, I'm going to have to count per minute how many times these jellyfish are pulsating. And then I'll count this one and that one and that one, and have a sample size however big I feel is important. And then compare my numbers. Are all of them pulsating at 12 pulsations per minute? If I went to a different species, would I see anything different? So this is a different kind of jellyfish. Am I seeing anything different here? So now, once I make one conclusion, I mean, to scientific thinkers, once you answer one question, then you typically think of far more. So once you say, that was my observation here, but I wonder if that applies to other species. I wonder what would happen if the temperature changed, et cetera. And so there was a question, but if you followed along with me, then you were being a scientific thinker. You were answering the question with data, all right? And it's amazing how long it took humanity to figure that out. When you have a question about the natural world, you measure the natural world and you answer it with data. That seems so obvious now, but for so long, people approach it where they already knew the right answer, or they tried to reason through it. So often today, when you ask, why, someone came to a certain conclusion, it's not based on data. We still don't do this as well as we could. So if you had a question about how amphibians mate, are they, mating in groups or not? Well, then you would have to watch amphibians mating. So here's all these pairs of American toads. Now, whatever you conclude about American toads, is that standard to amphibians? Well, here's a pair of mating newts. And so if you had a question, all right, about the natural world and made a hypothesis is a guess. I think that amphibians mate this way. You would then go out and measure the natural world, all right? And so, we watched the toads, we watched the newts, and we came to our answer. Now, science works in a lot of different ways. And so there, while we do have this standard scientific process where you ask a question about the natural world, then you make your guess. This is my hypothesis, I guess this. You design an experiment, etc. I'm sorry, did we just skip one? We did. There are actually different versions of this. I mean, science, is great. So some things are more descriptive. Some things are more descriptive. And so, for example, you could start off with, a question where, I think, tadpoles change from day 1 to day 2 of their life. Or you could simply say, I would like to describe the changes that tadpoles go from, day 1 to day 2. And then so, sometimes you can do the same experiment, with a question, or simply I'm going to note the changes. So here we see tadpoles on day 1. They've got these gills. I don't remember tadpoles having gills. They have this elongated body. You go to day 2, and now most of them are losing their gills. All right, their heads are getting fatter. They're getting more of the tadpole shape. You go to later days, then none of them have the external gills anymore. So whether you ask the question, what happens to the gills, from day 1 to day 2, or you approach it, I'm going to do more of a descriptive study. What are the changes that tadpoles undergo? You are, performing science. So science, is how we study the natural world. There's not one box. Every scientific study, fits into this. But for this, set of videos, let's just, do some standard ones and hit some big points. So once again, if you had a question, there's sponges on the seafloor. Do they affect fish? So for example, well, then one would study that. And then one say, all right, well, I don't know, let's take areas of the seafloor that don't have sponges, like this one, and then let's take sponges, and then maybe, I don't know, count fish around within, say, a 4-foot radius around, a sponge, and then we could pick random spots here and there. And do we see a difference in the number of fish in these videos? Now, one of the reasons I provided all these videos is that there are so many that you could then do your own scientific study. You could pause it at any point. You could pause a video and say, I'm going to pick a random spot here and there, and I'm going to count fish near a sponge, away from a sponge. And then I'm going to make my conclusion. This is how sponges affect fish, or this is how sponges interact with fish. The point is, you answered a question with data. So when we talk about coral reefs, all right, why are coral reefs important? I don't know, let's just look at the seafloor. All right, let's say, I don't know, count fish. So I start off with just the open seafloor, all right, and then you can look at, count fish. There's one, for example. Or then later we go to a coral reef, and then you can count fish. Now, you could count numbers of fish. You could count different species by the different colors, etc. And then if you had a question, how do coral reefs affect fish, populations? Now what you've done is you are basing your answer to the question, your conclusion, on data. You've actually observed coral reefs. That's what these videos, as I kind of incorporate some of the ideas of just scientific inquiry, that I would like you to practice. Once again, this is a skill. Like any skill, it gets better as we as we practice. And so this is meant to give you a bunch of chances to practice. Okay. Now, a couple of points. One point is that many scientific studies are going to compare two sites. All right. So we're going to compare, say, site 1 to site 2. And so we're going to be looking at the diversity of birds. All right. So here's a wetland. All right. And one wetland is going to be treated with an insecticide which kills mosquitoes. So whoever lives in this area decided that mosquitoes are a pest. You know, here's a pesticide. They say it's safe. All right. So we've gotten assurances from the manufacturer this is safe. But we don't know that. Once again, in science, we don't think we know anything. 100% at least. And so we want to study that. So here's site. One site is sprayed with the pesticide, one site isn't. And then we will look at birds. All right, now, a couple of things I'm going to develop over time is, one, you have to decide, well, how are you going to measure birds? So how am I going to count them? Am I counting numbers? Am I counting different species? Because that is what we call the dependent variable. There's two things that are varying here, or at least one thing definitely and one thing we think. That's our hypothesis. Is the one thing that we definitely know is that one site gets sprayed with insecticide and one doesn't. So that's a difference, all right? So there's a difference between site 1 and site 2. One was sprayed. Now that's a done deal, all right? And then so we know that it's definitely a difference. So this is what we call the independent variable. So the independent variable is that one site was sprayed with insecticide. The question then is, will that then be the cause of a difference in the number or diversity of birds? So that's what we call the dependent variable, because the dependent variable may then be caused, a difference here might be caused by a difference in the independent variable. So if you compare sprayed versus not sprayed, that may cause a difference in like little bird diversity, lots of bird diversity. And so many studies like this, the purpose of the study is we want to see the effect of the independent variable, all right, which we control. You spray here, you don't spray there, on the dependent variable that we don't control. This is something that we are then observing. So then as you practice thinking scientifically or thinking about these questions scientifically, then going back to some of the previous ones, the coral reef study, for example, we looked at ocean floor and coral reefs. All right, so there were two spots of the ocean floor that varied as to whether there were coral reefs there or not. That was the independent variable. The dependent variable that we were then measuring was, say, the number of fish. Does the number of fish vary on whether you have a reef or not? If you're comparing two different species of jellyfish and how often they pulsate, so the independent variable then becomes the different species. So you have species 1, species 2. Species 1 is in this tank, species 2 in this tank. That's, you know, a done deal. So that's your independent variable. And then the thing that you then measure is the number of pulsations per minute to see if the independent variable, the different species, then affects the number of pulsations per minute. So here's another one. So let's say we're going to go to two different rivers. Do they have the same kinds of wildlife? That's a good question. How does wildlife, how do fish, how do plants, how do bugs, how do whatever, how do they vary from one site to another? That's a good scientific question. Is there a difference in, say, biodiversity from here to here? Or is there a difference in the dominant plant from here to here? And so the independent variable would then be the two different sites. So site number 1, site number 2, all right, that's the independent variable. And then you then, however you measure life, that you're going to, you know, look at, you know, microscopic organisms, you're going to count fish, you're going to count birds or whatever, and then you're going to see, is there a difference? So that's you're answering the question with data. This is a scientific study. The data suggests that you look at the frequency of these plants or these fish at site 1, see it's similar or different to the ones at site 2. And so a bunch of these questions. Notice that all of these videos, if you're playing them, most of them have no sound, because the purpose was not to listen to a professor talking about science, but to give you the chance to do it. All right, so that you could ask your own question. You could count fish. You could freeze a frame and say, well, that would be a lot of fish, but like you could freeze a frame at any point and say, I'm going to count here versus count there, and then make a conclusion based on the data. All right, so in this next one, we could ask the same river 1 to river 2. So is there a difference between rivers? We could ask river to a lake. Is there a difference from a river to a lake? And study that. This last question, very important. With climate change, one of the things we're observing with coral is that it bleaches. It loses its pigment and can then die, shortly after. Well, how does bleached coral, which is happening more and more frequently with climate change, how does that affect? Well, so here you see all this white region. So here we start off with bleached coral. And then on the same reef, I then have healthy coral. And then you can compare for yourself. So don't read in a book, what's the difference between, what does bleached versus unbleached coral do? So now here you have healthy coral. And then do you notice a difference, given that it's in the same reef as the previous one? Okay. So the first question, which I hoped to just introduce, is the difference between an independent variable and a dependent variable, because very often when you set up a scientific study, you're comparing two things. This thing is different from this thing. And so very often there is an independent variable. So here's one site versus another site. Here's one species versus another species. Here's one frog versus another frog. All right. So I'm going to hypothesize that all spring peepers make the same number of peeps in a minute, or that spring peepers near the water are going to make more frequent vocalizations than those which are farther from water. All right, so my independent variable is one frog versus another, or frogs which are near water versus frogs which are far from water. And then the dependent variable is then the counting of the vocalizations. Okay, so that was one thing that I had hoped to just, bring the idea in, the idea of dependent and independent variables. Now, just a caution on that. It is important to minimize the differences. Now, this video, it explains this point. It's a little more on the genetics end, so you don't have to worry about the finer details here. But the point is, if you're going to say that I'm going to compare this to see if the independent variable has then caused a difference in the dependent variable, you have to try to minimize the variables. So for example, if I compared this river to this lake, and I'm looking at aquatic life, for example. So I'm going to say, all right, I'm going to sample this river, this lake, and I sampled this one at night and this one in the day. That's not a good scientific study, because there's one difference, all right, in the independent variable in the experiment, which we could call the experimental variable. So there was the site, the river versus the lake, but then there was also a difference of the time of day. So there were two experimental variables which varied. So there was, one was at day, one was at night. So if you see a difference, wow, there's different wildlife, in spot 1 versus spot 2. Well, something caused a difference, but what was it? Was it, say, the time of day, or was it, river versus lake? So there's a difference, but now you don't know what caused it, simply because that there were too many variables. A good experimental design has to minimize the number of variables. So here's, for example, when we try to understand opiate effects on the brain and addiction and et cetera. We're often doing animal studies, say with mutant mice. But once again, you don't have to go into all of the finer details here. But now here's one of the key things. Let's say, I have generated a mouse with a mutation in the opioid receptor. And I'm going to study those and then because they are not getting the input from opiates because their receptor doesn't work. Now I've learned something about how opiates work in the brain. But that's making the assumption that these two mice are equal in every way, except for this one doesn't have this one protein in the brain, an opiate receptor. Now, that's what you want, but you have to prove that. And so when you read a scientific study, they'll go to great lengths of all of these, experiments where they just establish that they were the same. Because if you're going to say, there's a difference in how they perceive, after we intoxicated one mouse, for example. But then you could ask, well, wait a minute, do they differ in other ways? Like, for example, do they now differ in how they perceive pain? All right, so if their perception of pain is different, then the difference that you notice might not just be, the opiate that you gave to one mouse and not, the other, but it might be this other genetic difference between them. So when you read scientific literature, I'll end with just kind of review of that. Very often they'll go into detail on how they minimize other variables. Going back to that study, if I'm sampling the river from the top of the water and sampling the lake from the bottom of the water, well, that's a problem because now I've got two differences, river versus lake, and then surface versus deep water. And so you would want to sample surface to surface or deep water to deep water so that the only variable was river versus lake. So that in an experimental design, we often would want to then minimize other variables as best we could. So perform observations on the same day or in the same season or in the same region or at the same time of day. Observations are always made at 9 o'clock, et cetera. We're always counting the same way. And so a good experiment, if you're going to try to say it was a change in the independent variable that caused this variation in the dependent variable, then the independent variable can be the only difference between group 1 and group 2. If there's too many differences, then if the dependent variable changes, you don't know which of the independent variable differences were the ones which caused that. Okay. So independent versus dependent variable, that was one of the topics I'd hoped to bring across. A couple other quick ones. Scientific questions, can vary in how deep they are. So some questions are called more descriptive. All right. So you could ask, for example, when ducks migrate, are they always traveling in groups of the same size? I mean, do the V formations, do they always have 20 ducks in them? Or do they vary in size? All right. That's a descriptive question. You can say, they vary. So some have, this is the minimum, this is the maximum, here's the average. You're describing something. You could then also ask an explanatory question. Why do they vary in size? So for example, are they more likely to be in larger groups in sunny days and smaller groups in stormy days? Do some species tend to form big groups and some species tend to form small groups? And so that's more, that could lead us then to more explanatory. Why is that? What's the advantage to that? And what are the factors which influence that? So here's more on corals. And so in, I think the next video, we could ask, are fish more likely to be around corals at a coral reef? And so there could be a descriptive answer, yes. So fish are more likely, look at all of these fish in the coral reef. So yes, the descriptive question, we're more likely to find fish here. But then a follow-up question is, why is that? And that may take further study. So for example, I don't want to tell you, what I think some of the answers are, because then you could study that. So you could now watch, all right? So here we are observing what fishes are doing in the coral reef. And so here, um, and we see one fish, and we can observe what it does with the coral. And I'm sorry if this is glitching a little bit, zoom in the internet, slow each other down. But you can watch what this fish does. You can watch in the next video clips, and then you could try to come up with an answer of why are fish here? What's explaining this? What is the adaptive value? So a lot of descriptive questions are measuring simpler things like how many of this, all right, and what's this, what's the range of this? Where does it migrate to in the winter, et cetera? And then more explanatory questions ask to why is that an advantage? Why are the organisms, doing it? What factors have, encouraged this behavior, et cetera? So there are different types of scientific questions. Some descriptive questions are simpler. Then others are more explanatory, getting more at the root causes. Finally, not only is one of the goals that perhaps you could now start looking at nature more scientifically or in a scientific way, whether this be your garden or the birds that come to your feeder, or maybe you study science and start asking questions. All right, so how would I get an answer to this? Let me set up a study where there's an independent variable, a dependent variable. How would I measure the dependent variable? How would I minimize other variables? How often would I repeat this to get the data that I wanted? Not only could you do that, but now because we have so many questions, obviously you can't investigate everything. We have questions about coronavirus. Certainly we do at the time that I'm filming this. We have questions about species going in danger of extinction. We have questions about climate change and coral reefs. So how do we answer this? Well, there's all of this literature out there. And so then hopefully then you can start looking at, some of these. So for example, here's, an interesting source of information here as well. But then a lot of sources of information are scientific studies published in refereed scientific journals. So here you have, experts in the field. They have published in a refereed scientific journal. So for example, if I had a blog, all right, if I have a web page, I could publish anything that I wanted to. I could say anything I wanted. There's a very low bar potentially of proofreading this or of error checking this. But for example, one would not publish in a scientific journal without experts weighing in and saying, wait, no, you didn't have a control group. You have too many variables. Your independent variable wasn't the only variable, et cetera. You couldn't publish this unless it was a good, high quality work. And so you should be able to now look at different sources of information and say, ooh, this looks like a scientific journal, published by, experts, et cetera. If you ever wanted to search for this or read this, very often in the citation, there's the authors, there's the title, there is the journal where it was published, there's the year that it was published, the pages, et cetera. If you start to look at some of these, I think you can come to even just reading the titles. Okay, a feathered dinosaur tail with primitive plumage. If someone were to ask, is this more of an explanatory thing or a descriptive thing? This seems to just be more describing. All right, so describing something that was found. So very often, a lot of scientific sources, it'll just this is how much ice is currently in the Arctic Ocean, and one could compare that from month to month or from year to year, et cetera. So that would be more of a descriptive item, where then some papers are now written more with the sense of an explanatory, why are we seeing a shift in rattlesnake behavior? All right, and then so is, once again, hopefully you are interested in science and biology to the point where you actually research this, not necessarily just on a website or a Wikipedia, although very good information can be found there, but then also the places where scientific study is published. All right, and then you could look at the materials and methods and say, oh, how did they limit their the variables? How often did they measure this, et cetera? Now, there are certainly scientific journals that you need to have a subscription for. So maybe your college, would have some. But certainly the proceeding of the National Academy of Sciences USA is for free online. This is the most prestigious scientific, organization in the United States. So there's a publication you can have right there. And many journals will then have older versions of their work online. Now, once again, this is just a precursor to science students, because the future of science is not exaggerating or not trying to, it's in your hands, all right? Because we are still learning about the natural world. Some of what we think we know isn't as correct as it could be. We need to learn more. So hopefully you contribute to that, maybe in studies that you do, or maybe, then you just become a more informed reader, seeking out those sources which are doing scientific research, where the experts are publishing. Because so many other sources, now you could have opinion and unsupported, claims play a greater role. And so as we try to understand the natural world, scientific inquiry needs to be at the root of that. And then hopefully, then this is a skill which we all could practice. I certainly need to work on, but hopefully, we all practice. How, do I better base my conclusions on observations of the natural world? How do we better test the natural world? And so the videos here, a lot of them, like I said, have no sound, with the idea that this was simply now your chance to actually observe, live video of the natural world. Make your own questions, all right? And maybe say, I could design a study, that would go this way. This is how I would minimize variables, etc.